So, first off, we can say that in 1859, Charles Darwin published a little thing called On the Origin of Species. And somewhat ironically, from that point forward, science really hasn't had a good idea of what species are. So we speed up to 1942, when a man named Ernst Mayr came up with the biological species concept. His concept stated that species are groups of actually or potentially interbreeding natural populations which are reproductively isolated from other such groups. Now, the key to this concept was reproductive isolation. What is reproductive isolation? It is the results of a multitude of mechanisms which prevent gene flow from one population to any other populations. Those populations are considered species. So scientists being scientists, they take this concept and they want to apply it to other organisms. So they apply it to birds, fish, insects, and frogs. They come back with two basic conclusions. One, Ernst is pretty much right. Species mate with like species. Duh, right? But the second conclusion is that he's not always right. There are times when these mechanisms break down in some way, and a species will mate with another species. And this can have catastrophic results. You have offspring that are inviable, and if they're viable, they're sterile. So in either way, you have zero. You have genetic zero, a dead end for that individual in that population if it spreads out. So in 1960, a guy named Fouquet proposed a set of isolating mechanisms specific for anurans, for frogs. I will go through a few of them that are set up for my study. There were more than these. The first group were anti-mating mechanisms. They are, as they state, anti-mating. There is no way for a species to mate with another species with these mechanisms. Underneath these are temporal isolation. If a group of species separate themselves out over time, they can't meet up with each other, no mating mistakes. Underneath these, more specifically, is seasonal. If you separate yourselves out over seasons, then yet again, you can't meet up, you can't make mistakes. Another set of mechanisms are courtship mechanisms. They are also self-explanatory. Underneath them is a little bit less explanatory. Ethological isolation. What that comes down to is behaviors. Behaviors that are based in courtship that are specific to species. So if this species has this set of behaviors during courtship, they will not be recognized by a different species. For frogs, everybody understands one of them. Calls. You go out to a pond, you go out to a lake, you go out to a stream, spring, summer, you hear lots of calls. Each of those calls is a species, and the females don't recognize species that are not their own. The other, that is a little bit less known, was proposed by Duhlmann. These are male call site preferences. If a species prefers to call from this area and not this area, and the females kind of recognize that, which is possible through female expectation. The females may recognize where they're going, where the males should be, or where they're hearing the call and going towards that area, then they should not meet up with other species. It may improve call propagation. Maybe by the male calling from this area, his call can get across easier, across the environment in such a way that the female catches it better, receives it better. What is the advantage of studying frog calls to begin with? They're species specific. As I said, if you hear a species, you know it. It will make that same call over and over, which is the next advantage, stereotyped. If you hear it, you can know it's going to make that same call again and again and again and again and again. And if you come back the next night, it's going to make that same call again and again and again and again, because it definitely wants to find a female. Small repertoire. You can have a female, you have males that have very, very small numbers of calls. They have about 3 calls in general. You have an advertisement call to find a female, or to just let males know that you're out, now you know that you're there. You have an aggressive call, which males give if a male gets too close to another male. And you have a release call. That's more if a male jumps on another male in an intimate fashion. Then the first male wants the other male to know, hey man, I'm a man. Back off, back off. And so they have very few calls. There's a compact multi-species assemblages. Basically what that means is that at a small pond you can have lots of species. If you're studying birds, you may have to go hunting them down. I mean, they fly away. Frogs are not going to go anywhere. You have a decent amount of species. They're staying right there in your little pond, and you can just move around and sample them at will and compare them as you will. So it makes it easier. There are other attributes besides just the call. You go out to find the call, but while you're looking at the call, you can find out phenology. Phenology is a snazzy word for when they're calling, which correlates back to seasonal phenology, when they're calling during the season. So you can get that, and you get where they're calling. That goes back to call site preference. So you can get all that while you're studying just the call itself. Primary questions for this study were: to what extent do calling phenology, when they're calling, call site preference, where they're calling from, and the call itself, the characteristics of the call itself, what extent do these three separately and together minimize acoustic interference among the species and maximize their isolation from each other? I also wanted to know, what role does size and air temperature play in the acoustic variation among the species at this community? So, what did I use? Where did I go? Materials and methods. The study site was a place called Frog Pond. This is not a coincidence. But it is, or was, tragically, a 2-acre seasonal wetland outside White Plains. If you're around the area, you understand White Plains. If you know, Frog Pond itself is about a 25-minute drive from here going down Whitescap Road. You get to it. 2010. It's nice, it's lush, it's got water. This is it two weeks ago. There is no pond now. And my wife and I have taken to calling it Bug Field instead of Frog Pond, which is kind of tragic actually, but we're hoping that it comes back in some way. At any rate, we move on. The study period was from March to July of 2010, and from February to May 21st of 2011 for a total of 37 study days. May 21st is a very specific day because that was the last day that a frog was heard. After that day, there was not enough water for them to motivate calling, and then it dried up completely. Data collection. For ecological data, phenology specifically, you just take notes on what you hear out there. Every time I go out there, I just write down what I heard. It's very simple. For individual recordings, I would use a digital recorder, about the same size as this. And I would get about ½ a meter or less to an individual frog, and I would record about 10 calls from anything that was out there and everything. I tried to get as many species every night as I could, and that's just what I did. Along with the calls, 10 calls, I'd record where they were calling from. I'd try to capture them if I could. If I could was a really good question, if I could. Measure them once I caught them, snout to vent length, and I'd release them. I would also gather temperature data at the beginning of the night and at the end of the night. Data analysis. I analyzed the ecological data, like I said, the phenology data. I analyzed it for month by month and seasonal phenology over time, and I'd analyze it for species richness. Richness is just an ecological term for what species are out there, how many species. How many species is richness. So I'd analyze it for species richness and how species accumulated over the seasons. For this presentation in time, I'm only going to present the phenology data. Acoustic analysis. All parameter measurements were taken from waveform and spectrogram analyses using a software called Raven 1.1, acoustic software. It's Cornell. And 1 to 5 call groups. I told you that I recorded 10 call groups. So I choose 1 to 5 of those, the best 1 to 5. You get background noise and things, and so you choose the best ones. I choose 1 to 5 call groups per individual and measure the following things. I measure dominant frequency. Don't worry if you can't keep up with this. There's a picture coming up that'll help you out. Dominant frequency in hertz, call duration in seconds, low frequency peak. Low frequency peak needs a little bit of explanation. These two frogs, Hallucinaria halogradiosa. This is the green tree frog, this is the barking tree frog. They have 2 peaks in their call. If you looked at a spectrogram, you'd see 2 peaks, a low frequency and a high frequency in their call. Past literature has shown over and over that females recognize the low frequency. So, for my study, I took the low frequency and used it as a substitute for any analyses that needed dominant frequency. So anywhere you see dominant frequency in results or anything, you can say that I used the low frequency for those two frogs. I also took pulse rate. I measured pulse rate. There were only two frogs. This is the Cope's gray tree frog, and this is the upland chorus frog. They are the only two frogs in this community that had pulsed or trilled calls. You will be able to hear this in a minute, because I have snazzy sound. I only used the pulse rate for temperature regression, temperature study. This is what I saw over 1,000 times during my study as I measured things. This is an example of a green tree frog. This is their call, and what I would have you pay attention is this top part first. This is what's going on in seconds. This is the time of the recording. This is the frequency of what's going on in this part of the recording. To get duration, just measure from here to here. Now I would have you pay attention to this red line. This red line represents a slice of this recording, which is all of this. All of this is in this one line. This is hertz, this is kilohertz, this is frequency in kilohertz, and this is the intensity of the sound in decibels, going up this way. So, literature has shown, over and over and over, that this is the low frequency peak. This is a good range for the low frequency peak of the green tree frog. And this is a good range for the high frequency peak of the green tree frog. If this were most other frogs in the study, we would be using this, because this is the highest intensity, the highest decibel, so it is the dominant frequency. But like I said, for this frog, we use the low frequency peak. So that's just kind of an idea of how I measured every time for over 1,000 times for different frogs. I had to analyze the data set itself. This is not always necessary, but I felt it was necessary and there was good reason. You have to I analyzed the data set for statistical assumptions. Well, everybody knows what an assumption is, right? So, sometimes bad. But you analyze the statistical assumptions, and I analyzed them for three things: normality, variance equality, and covariance equality. Like I said, these are usually assumptions, but I'm glad I analyzed them because for my data set, these assumptions were not met. And that justified why I had to use non-parametric statistical strategies for this data set. Normally they like you to use parametric, but if it's right, and non-parametric seem better for this step. The statistical tests themselves, univariate analyses. Basically what that means is that I did tests on dominant frequency and call duration separately. And on both of those parameters, I did a non-parametric Kruskal-Wallis test, a parametric ANOVA, and a post hoc Mann-Whitney pairwise comparison. Basically what I'm trying to get at there is, are there significant differences? Why, come on in, Dr. Mead. Are there statistical significance? Are there differences among this community in dominant frequency and call duration separately? For multivariate analysis, this combines the two. I said univariate looks for differences in both of those separately. Multivariate combines the two and asks, are there differences in the community when you combine the two parameters? And so I did a non-parametric MANOVA and a post hoc multivariate pairwise comparison. I also did a quadratic discriminant analysis. Science is mainly used to thinking in linear, but quadratic was better for my study because quadratic was the non-parametric method of linear discriminant. So what it does, it asks a simple question. Are these call parameters, that dominant frequency plus call duration, are these parameters discriminating characteristics for the species in this community? Do they discriminate? Can a species be categorized as itself just according to its calls? Are there going to be any mistakes? Significance level of tests. We're all at alpha equals 0.05. That means for probability levels, I need a 0.05 or less to be significant results in my study. I did a regression for certain things. I did a regression for snout bent length versus dominant frequency for each species in the community. and I did an ambient air temperature versus respective call parameters for each species in the community. I just wanted to see, I want to know if snout vent length and ambient air temperature has any effect on dominant frequency or different call parameters. For this presentation, I'm only going to present the snout vent length regression. I used a PASS 2.16 using for all univariate and multivariate analyses, and I used XLStat Pro 2012 used for QDAs and regression analyses and the visualizations. So results. What did I find out? Field observations. All right? You're about to hear calls. It could be a little loud, but it could be fun. But nonetheless, I'd have you pay attention to the calls and try to hold on to like what you hear, how high things are, how low things are, and I'll give you some hints as you go along. This is a barking tree frog. This is a green tree frog. You will probably have heard some of these frogs maybe in your backyard or your parents' backyard or just around town. This is a Coke's gray tree frog. This is a cricket frog. This is a spring peeper. This is an upland chorus frog. These are the ranids. That is the family they belong to. The last slide was full of hylids. Those are families. So for ranids, we had the green frog. He was recorded, but I could never actually capture him. He was in the grass and he was annoying. But nonetheless, wait for it, wait for it. That is his call. This is a bullfrog. He was not recorded or captured because at my site there was only one ever. And I could not get close enough nor find him. So there was one. This is a southern leopard frog. He was not recorded or captured either because they laughed at me. If you can hear they do laugh, that is their call, they laugh. And they laughed at me and there were 50 million of them in front of me, I just could never get to them. Que sera. And this is a microhylid. This is a little bitty toad. That is full size. That is a narrow-mouthed toad. He was recorded but not captured because he was in some mud and there was about 7 of them, and we won't go into that because that's frustrating too. But wait on it. That is a narrow-mouthed toad. So, summary of sample sizes. This side, for those who are interested, would be the N. This would be N for sample size for statistics. This was the number of individuals recorded. This was the total number of call groups for those individuals. And like I said, I took 1 to 5 of 10, and I took the mean of that. This is the total number recorded as well. This is the total number that were recorded and captured for measurement. So some of those I said I recorded but didn't capture, so that kind of works out there. Calling phenology. When were they calling? In 2010, the hylids, that first slide, the smaller frog, if you will. The smaller frogs dominated the calling during the early part of the study with the spring peeper. Yep, he was dominating the calling, and he began and ended before all the others. Or to be more specific, he began before everybody else, and then others and he ended while they were beginning. You'll see in a picture in a second. So these are the guys that, these are the other hylids that called during 2010. The ranids, the larger frogs, the second slide, they followed the season progressed after these guys, and then the little microhylid, he followed closely with the ranids. And this is kind of how that plays out. We have the peeper down at the bottom. He starts before everybody else. They kind of follow in on him, but he shuts down. And all that's left are these hylid species, and then right here you start the ranid species that follow in, and then you have the small gastrophryne, you have the small narrow-mouthed toad. But these guys pretty much overlap if you get right down to it. I really can't count that guy because that was that single bullfrog, and I really don't know what to do with him. He's not even really supposed to be there, I think. But at any rate, you have a lot of overlap, but these guys shut down. In 2011, calling patterns were distinct from 2010. We had an early inclusion of the leopard frog and the chorus frog. They were not seen in 2010, and so that was kind of cool. We had a late exclusion of the one bullfrog and the narrow-mouthed toad. They were never seen again. Many species called later and ended earlier in 2011-2010, but that makes good sense because by May 21st, there was no rain and there was no water, so so they all shut down. 2011 season is a really hard one to look at, but at any rate, it is what it is. This is how it worked out pretty much. The peepers and the leopard frogs and the upland chorus frogs, they all called together, and then these guys call at the end, and then everybody shuts down on May 21st pretty much. Crepitans, the cricket frog, was the last frog that I heard. Call site preferences. Where are they calling from? We have 3 general locations. You have frogs, the barking tree frog and the green frog, call primarily from floating or partially submerged areas. You have peepers, chorus frogs, cricket frogs, narrow-mouthed toads, calling from ground level or waterline. This would have just been waterline, but in 2010, when the water was highest, I'd say I was in full waders and it would be up to about here in Frog Pond. And so I got my digital thing out here and trying to keep from slipping and killing myself. And it was fun, and I'm glad nobody's recorded that. But nonetheless, they were all on the ground. They were all inside the shore, on the grass. And then in 2011, when the water was down to almost nothing, they were all inside the pond calling from emergent vegetation that was just popping up. And so that's actually kind of interesting. And along with that, fairy animal, like I said, was not even there in 2010 when the water was so high. But when the water was really low, the chorus frogs were there. So that maybe that plays out as an environmental factor for chorus frogs. We had green tree frogs and Coke's gray tree frogs who preferred to call up above the water. I'll get more into that later. You have an analysis I decided to do of my own. I analyzed the dominant frequency, which is this, versus the species. And I found that small species, all my smallest, highest frequency species, like to call from lower. They like to call at emergent vegetation or waterline or ground level. My 2 medium frequency species like to call from up above. They like more arboreal, they like to be elevated. And the 2 largest species with the lowest frequencies like to call submerged or floating. And there may be something there. There may be some localization, because the whole purpose is to get females. The whole purpose of the calls at all is to get females. So there may be some localization there, something that females can cue in. If a female goes towards these areas, and as they get closer to these areas, maybe they can localize and pinpoint their species call as they get closer. I don't know, but it's interesting. Vocal and size descriptions for these species. This is a lot of numbers, and it's okay. You don't have to put it all together, but I can help a little bit. Just so you know, we have dominant frequency, call duration, pulse rate, snout vent length, and the species. What you can get out of this is that we had species that had relatively long calls. You have species that have relatively short calls. 0.1, 0.17, 0.16, 0.14. You have species that call it relatively high frequencies. You have species that call it medium frequencies. And you have species that call it low frequencies. Now why that is important is because, in general, the small frogs produce high frequencies. This U means that it was uncaptured. But from historics and from field guides, a narrow-mouthed toad is about 35 millimeters. So he fits really well into that category. As you get lower in frequency, you get a little bit bigger in size. As you get lower in frequency, you get even bigger in size. This U is for the green frog, which I was uncaptured, but they can get to about 85 or 100 millimeters. They are the largest frog there. And so they fit really well into the low, large frog category. And that's important later. So community parameter analysis for univariate data for dominant frequency and call duration by themselves. This is the non-parametric versus the parametric. The interesting thing about that is that while I understand that the data is non-parametric in general, to do both of them was really important because both of them turned out the same. They both showed highly significant statistically differences in both of those respectively. So the community has significant differences among the species in dominant frequency and significant differences in call duration. For the ANOVA, it came with a little bit extra. It came with a little bit extra to analyze. There is 93% of the variation in this community comes from dominant frequency, while 82% of the variation in the community, in the differences in the community, come from call duration. So from this we can say the dominant frequency holds a little bit higher ground in importance as far as the parameters of these calls. Dominant frequency holds a little bit higher importance than the call duration in recognition by females. This is a comparison test. What you're seeing here, just so you know, these are species and these are the same species. This is a redundancy chart. These symbols correlate with these in the parentheses. These are the non-significant measurements. What this says is that Pseudacris crucifer is similar in dominant frequency to the narrow-mouthed toad. Pseudacris crucifer is similar in dominant frequency to the upland chorus frog, and the upland chorus frog is similar in dominant frequency to the narrow-mouthed toad. I would have you hold on, if you can, to these three stats for a little bit later, because they're kind of important. This is more of a visualization of what I just gave. Narrow-mouthed toad, peeper, peeper, upland chorus, narrow-mouthed toad, following across here. This is a univariate, just call duration. So what we have here is a little bit, it shows that call duration has a little bit more overlap. So we have Coke's gray tree frogs having similar call durations to upland chorus frogs. Upland chorus frogs have similar call durations to narrow-mouthed toads. Green tree frogs have similar call durations to barking tree frogs. And green frogs have similar call durations to barking tree frogs. So there's a little bit more overlap as far as call duration goes in this community. Visualization of that as well. We have the copes and the upland. We have the narrow-mouth and the upland and the chorus frog. We have the green tree frog and the barking tree frog. Well, let me get back. This is the, yeah. And this is the green barking tree frog and the green frog. This is the cricket frog. He had to be placed all by his lonesome because if you'll notice how this scale in seconds is going, you have 0 to 1.6 just for this scale. This scale starts at 0 and goes to 16. The cricket frog's calls are so long that if I place that in this scale, all of these will be squished down to here and you wouldn't be able to see any differences. You wouldn't be able to see anything. So multivariate. So how does the community look if you combine the two parameters together? Very much the same. There are significant differences in the community as a whole if you combine both parameters together. The pairwise test using both parameters together looks very similar. Actually, it looks identical to dominant frequency. You have the same comparisons and the same overlap between the same species if you use dominant frequency or if you use both parameters. If you do look at QDA, if you look at the discriminant analysis, this is looking at predicting species. What this program shows is that 94% of the time the program was able to properly predict the calls with the species. But there were some problems. There are problem species that overlap. You have, sometimes you have a narrow-mouthed toad, which was predicted as a Coke's gray tree frog, and sometimes a narrow-mouthed toad was predicted as an upland chorus frog. You have a green frog, which was half the time predicted as a barking tree frog. And you had an upland chorus frog, which was predicted some of the time as a narrow-mouthed toad, and some of the time as a Coke's gray tree frog. Now, I showed you earlier how the size matched up with the snout vent lengths. Well, this is more of a straightforward view. We have the smallest at 23 millimeters and the largest at 57 millimeters. You have the highest at 3.7 kilohertz and the lowest at 0.444 kilohertz. As you get smaller in size, you get higher in frequency. And it's a fairly impressive difference because you have 99%, about 99% of the variation in dominant frequency is associated directly with how big the frog is, which is pretty good stuff. So let's discuss it. Let's just kind of discuss it and go back and do a synopsis of things. For temporal isolation, there were two general groups. The late winter breeders And spring callers, peepers, forest frogs, leopard frogs, and mid-spring summer callers. Everybody else. For ethological isolation, for the advertisement calls, you have 3 groups. You have those that call from the water line and ground level, and you have phones that are going off, and that's okay. And you have the arboreal frogs, and then you have the aquatic floating frogs. Now, even though you have these groups, there's some differences. The narrow-mouthed toad was never found inside the pond. These other 3 only call from inside the pond. The narrow-mouthed toads always call from muddy areas just around the edges of the pond. So it would be easier for the females to get to them, to understand that they're there and not inside the pond. The Coates gray tree frog and the Cinerea were never, and the green tree frog were never found together. Gray tree frogs always called from trees, period. Just from trees. Always from trees, never found anywhere else. While the green tree frogs had more types of sites. They were always above the water, but they would choose bushes, they would choose bulrush stems, they would choose trees, they would choose anything that was above the water. They were a lot more generalist in their types of sites they'd call from. Advertisement calls. There are two call parameters. Acoustic variation, discriminant analysis, using dominant frequency and call duration suggests that effective acoustic separation exists for most of the species in this community. Dominant frequency being primary and call duration being secondary, calls should be sufficient for majority of females to recognize their own species aside from other species. And there were problem species, and we're going to go through those in a minute. We have peepers versus narrow-mouthed toads, chorus frogs versus narrow-mouthed toads, green frogs versus barking tree frogs, and peepers versus upland chorus frogs. And we'll go through those in this, the additive effects of these isolation mechanisms. So, peepers and narrow-mouthed toads, they apparently have problems, but they never see each other. Seasonal isolation, I was saying near the beginning, they don't see each other. You have the peepers calling in the winter, or in the winter and early spring. Narrow-mouthed toads are summer callers, so they don't have to worry about making mistakes in mating. Upland chorus frogs, narrow-mouthed toads, same issue, seasonal isolation. They won't see each other to make mistakes. The green frog and barking tree frog, right off the bat, you have a major size issue. They're just, you have an 85 versus 100 millimeter versus about a 40 millimeter frog. There's just mechanical differences there that won't allow mating. And you have interfamilial differences, not just intergeneric. I mean, you have Lithobates versus Hyla, and those are genus issues. But you're dealing with ranids versus hylids, and those differences may result in courting behavior in separation in courting behavior of females that I can't account for. But it makes sense that they would be there. And there's a call envelope. It's called a call envelope at any rate. When you heard the frogs, there was a difference between a barking tree frog. You could hear boop, 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 and then you had to wait a little bit to hear boop, boop. They're similar frequency, and they're similar in duration, but there's a tangible difference in what you're hearing. And that difference is considered a call envelope, and those differences have got to be a lot more significant for a female listening. The peepers and the chorus frogs, similar phenology. They called from the same season. They do call in the same season. Similar call site preferences in 2011, but not 2010. In 2010, the upland chorus frog wasn't even there. The water was so high. It's been my experience, and I have anecdotal evidence that they mainly call from little ditches, and they'll call from puddles. They like lower water. They're much more adapted for lower water depths for calling and breeding. And so it's possible that in 2010, whenever the water is too high, they're just not there. You wouldn't see them there, so it wouldn't have any competition. As I said, it's possibly precipitation based. They also have very different temporal structures. You heard versus I have no spit. At any rate, you have temporal issues, and you have and so there should be a major difference there. But having said and shown that, there's still been shown to be interbreeding between those two species. If the variables are just right, then there has been shown to be interbreeding between them. But the offspring shown to be inviolable. So that's still pretty good isolation. Conclusions, advertisement calls shown to be primary means of species isolation for most of the species temporally present in this study. Dominant frequency seems to be the major player for this isolation. For temporal separation, it's important in reducing acoustic interference, and it isolates certain species that were in this in the community. You had, like I showed, the peepers versus the narrow-mouthed toads, and the peepers and the coarse frogs versus the narrow-mouthed toads. There was a Couch in 2000, Jimmy Couch, who basically stayed the night at Frog Pond. He spent the night there, and he recorded calls over the course of an entire night and found that there was a diel separation. So over the course of a night, you have different frogs that kind of take the stage, and they'll go down, and another species or two will take the stage and go down. So they're separating the night. So maybe that is another level of separation. Spatial separation, where they're calling from. Important in reducing acoustic interference. May have acoustic localizing effect on listening females. That was the possibility of that dominant frequency versus species. And it's been shown historically to have some isolating effect on certain species, specifically the green tree frog and the barking tree frog. It was shown that if you cut down the arboreal, if you cut down the elevated perches, then you increase the contact that these two frogs had. And if you increase the contact, then you get more hybridization and more mating between these two. So they shouldn't meet at all based on spatial separation. But if you mess with things, then you mess with their mating strategies. Mechanisms combined, with all three combined, isolation among the species in this community is maximized under stable environmental conditions. And as I said, levels of interspecific isolation may be affected by stochastic. That's random changes. By random changes in the environment. You get more rain one year, then you cut out one species that really likes low water. If you lower that water, then you bring a species in that would not normally be there. If you cut out some part of the environment, then you increase contact between species that would not normally have that contact. And there's a large amount of variation can be attributed to interspecific size differences. 99% of that variation specifically. I would like to thank George Klein, who is Dr. Frog, and he, without his guidance and frogness, would, well, it is what it is. And I thank him a lot for his guidance. I'd like to thank Dr. Murdoch and Dr. Meade. They helped out with some of give me a refreshing look at my thesis at times and let me see some things in the making of it that are nice. Nicole, I think my wife, I would actually be typing at times, and she would be, she had my recorder, and she'd be on Word managing my data. She put like sound bit length on Word files, and then she'd hand it to me. And so she actually did a significant amount to help with this thing. Dr. Jan Case over in math and science, she helped out with some statistical stuff. Tom Allred helped out with some software. Biology department for being biology department. And Jacksonville State University became because, well, that's where I went to school. And I'd thank Dr. Frank A. Romano. This was his last words to me at his roast. Right at the damn table. And it is written, sir.
You mentioned for most of them you did the high frequency or the dominant frequency, and then for two of them you did the lower. Why was that again? Because
past literature has shown that females, for those two species that I did the low, the females cue in on that low instead of the high. And it just didn't seem to make sense at the time to include the one that the females don't care about. Because that's the whole point of them, is the females need to care. And that's the point of life, isn't it? Females need to care. That's right. Or cricket frogs. Or crickets.
Okay, let me see. What did I write down?
I have a delay. Go ahead. Yeah.
You were talking about frequencies of the calls.
Okay.
And I guess what I started thinking a little bit about was, you know, the life histories of these frogs, and I know, frogs disperse out, they come to the frog pond during the breeding season and that, but the frequencies related maybe to what their home ranges are during the breeding seasons. If females are closer, you don't have to do, you can do deeper frequencies as opposed to higher frequencies. Higher frequencies travel further, right?
physically.
So if it could correlate to home range or not. I guess that's what I started thinking about whether or not the call frequencies could be related to how far the females are expected to be from the male. I guess that's what I was thinking.
Well, actually, how far they're traveling is a decent question. How close they are to the pond to begin with is a decent question.
And I don't know.
I don't have a great grasp of where the females are or how close they already are. They could be coming at the same point. The males could be right there at the pond, just waiting for the rain, and the females may be, I mean, they could be right there at the pond too. I mean, or at least within proximity. Different frogs are probably closer at different times. I don't know the home range of different frogs, how far out a frog will go.
Some of that might be where you measured or recorded the call from. Frog high frequencies tend to drop off in intensity at a rate of, you double the distance and you cut the intensity in half. So even small variations in whether I was in 1 meter versus a half a meter away from those frogs are going to have an impact on the intensity that's recorded, especially at the higher frequencies. So just a little bit of variation in where you got that might account for why there's more energy in one of those peaks when you'd expect it to be at a lower peak. If we'd have been able to...
Did that have anything to do with
It,
Yeah.
yeah, it can. I mean, it could. I mean, I don't know. I didn't know if it was a valid question or not. I was just thinking about it. I was thinking about what you were looking at there, and I was going, well, it seemed kind of a... So I was thinking how the frog calls were transmitted over the surface of the water is what I was thinking. And that could be related to position and where they're
related
at.
to frequency of the call too.
Right.
Are males and females sexually dimorphic?
Yeah, well, in general, females are a lot larger. Some frogs have females that are more sexually dimorphic than others, but size is your main player. That's almost your only player is usually size.
So did you collect females as well as males?
No, this was sheer acoustics. Just the males talking to females. Didn't get any idea of how successful their calls were. So that's a whole nother study. It'd be nice if their waters do it, but no water.
Well, since you brought that up, that's one of the things I thought about wasn't part of your paper there, but wouldn't it be neat, if there was water in the pond, to go through and do your frog call analysis, and then you go through and sample the tadpoles that are present in the pond and use that kind of as a measure of who was successful, whom, at least in the species range.
John, right? That's not even a hypothesis now, though, unless they get water.
But yeah, granted, water was the limiting factor.